Regulation of gene action. Gene regulation or genetic regulation is the controlled expression of genes in response to external and internal signals. Genes contain the coded information necessary for the synthesis of enzymes and proteins. All at the same time, most genes do not operate all the time to produce the maximum amount of enzymes or other proteins. They are active only when their products are needed for the cell. All the other time, they remain switched off. So, a living cell will never get overloaded with unnecessary or non-essential proteins and enzymes or with an excess of essential ones. Constitutive genes and regulated genes. Some genes are always active since their products are needed all the time. This results in the continuous production of the required molecules such as ribosomal components, tRNAs, RNA polymerase, glycolytic enzymes, etc. Such continuously active genes whose products are needed always are called constitutive genes or housekeeping genes, and their continuous expression is called constitutive gene expression. But some other gene products are not required always but are required only under certain conditions. So, under normal conditions, such genes may remain repressed or turned off. They get de-repressed or turned down and become active only when a biological demand calls for their action or when their products are needed by the cell. Such genes, whose expression is periodically regulated and turned on and off in response to the needs of cell or organism, are generally called regulated genes. Then gene induction and gene expression. Inductive and repressive gene expression. The regulated genes whose expression can be induced or stimulated by specific molecular signals, agents, or factors are called inducible genes. The regulated genes whose expression can be induced or simultaneously stimulated by specific, not simultaneously, just stimulated by specific molecular signals, agents, or factors are called inducible genes. The process of their activation for expression is called induction of gene expression, and the regulatory substances which bring about gene induction are called gene inducers or gene activators. On the other hand, the genes which are inactivated or inhibited by some signals or agents are called repressible genes. The process of their inactivation is called gene repression, and the substances which bring about gene repression is called gene repressors. Then regulation of gene expression. Different gene products are synthesized in a cell in different amounts under different conditions. This means that the expression of genes is very precisely regulated in accordance with the structural and functional properties of biological demands of the cell. Regulation of gene expression is significant in two respects. It maintains a balanced amount of gene products and alters the rate of synthesis of some gene products to bring about structural and functional changes. In prokaryotes and eukaryotes, the expression of most genes is regulated. However, the molecular mechanism and the major manifestation or effects of gene regulation are different in both the groups. In both, gene expression is regulated mostly by the deregulation of transcription. However, the regulation of some genes occurs at the translational level. In eukaryotes, yet another mechanism also operates sometimes, namely the post-transcriptional processing of the primary mRNA transcript within the nucleus. In prokaryotes and eukaryotes, the expression of most genes is regulated. In prokaryotes and eukaryotes, the expression of most genes is regulated. However, the molecular mechanisms and the major manifestations or effects of gene regulation are different in both the groups. In both, gene expression is regulated mostly by the regulation of transcription. However, the regulation of some genes occur at the translational level. In eukaryotes, yet another mechanism also operates sometimes, namely the post-transcriptional processing of the primary mRNA transcript within the nucleus. The gene regulating systems of eukaryotes have maximum economy and efficiency to maximize the rate of cell multiplication. To some extent, this is true to unicellular eukaryotes also, such as unicellular fungi, algae, and protists. On the other hand, in multicellular eukaryotes, regulation of gene expression results in cell differentiation. Then the gene regulatory systems of prokaryotes have maximum economy and efficiency to maximize the rate of cell multiplication. To some extent, it is due to This is true to unicellular eukaryotes also, such as unicellular fungi, algae, and protists. On the other hand, in multicellular eukaryotes, regulation of gene expression results in cell differentiation. Then, positive and negative gene regulation. There are two main kinds of gene regulation, namely positive or inductive regulation by the induction of gene expression, and negative or repressive regulation by the inhibition or regression of gene expression. In positive regulation, the presence of a gene activator enhances gene expression and its absence retards gene expression. Conversely, in negative regulation, the presence of a gene repressor retards gene expression and its absence enhances gene expression. In fact, positive and negative gene regulation or induction and repression of gene expression are accomplished essentially by the same molecular machinery or mechanism. Then, regulation of gene expression in bacteria. Bacteria regulate the expression of their genes in order to control the amount of their gene products and also to conserve their energy and resources. They accomplish this feat in such a way that only those gene products that are necessary for the cell are produced, maintaining dynamic balance between the rate of their production and the rate of their degradation. Regulation of gene expression enables bacteria to respond to changes in the environment, particularly the presence or absence of nutrients. The rate of synthesis of gene products can be regulated in three possible ways, namely by regulating the gene rate of transcription. So these are regulated genes. Then gene induction and gene repression. Inductive and repressive gene expression. The regulated genes whose expression can be induced or stimulated by specific molecular signals, agents, or factors are called inducible genes. The process of their activation for expression is called induction of gene expression, and the regulatory substances which bring about gene induction are called gene inducers or gene activators. On the other hand, the genes which are inactivated or inhibited by some signals or agents are called repressible agents. The process of their inactivation is called gene repression, and the substance which bring about gene repression are called gene repressors. Then regulation of gene expression. Different gene products are synthesized in a cell in different amounts under different conditions. This means that the expression of gene is very precisely regulated in accordance with the structural and functional properties and biological demands of the cell. Regulation of gene expression is significant in two aspects. It maintains a balanced amount of products and alters the rate of synthesis of some gene products to bring about structural and functional changes. In prokaryotes and eukaryotes, the expression of most genes is regulated. However, the molecular mechanisms and the major manifestations or effects of gene regulation are different in both the groups. In both, gene expression is regulated mostly by the regulation of transcription. However, the regulation of some genes occurs at the translational level. In eukaryotes, certain other mechanism also operates sometimes, merely the post-transcriptional processing of primary mRNA transcript within the nucleus. The gene regulating systems of prokaryotes have maximum economy and efficiency to maximize the rate of cell multiplication. To some extent, this is due to unicellular eukaryotes, also such as unicellular fungi, algae, and protists. On the other hand, in multicellular eukaryotes, regulation of gene expression results in cell differentiation. Positive and negative gene regulation. So we learned there are two kinds of gene regulation, like positive and negative gene regulation. There are two main kinds of gene regulation, namely positive or inductive regulation by the induction of gene expression, and negative or repressive regulation by the inhibition or repression of gene expression. In positive regulation, the presence of a gene activator enhances gene expression. In positive regulation, the presence of a gene activator enhances gene expression and its absence retards gene expression. Conversely, in negative regulation, the presence of gene repressor retards gene expression and its absence enhances gene expression. In fact, positive and negative gene regulation or induction and repression of gene expression are accomplished essentially by the same molecular machinery or mechanism. Then regulation of gene expression in bacteria. Bacteria regulate the repression and expression of their genes in order to control the amount of their gene products and also to conserve their energy and resources. They accomplish this feat in such a way that only those gene products that are necessary for the cell are produced, maintaining a dynamic balance between the rate of their production and the rate of their degradation. Regulation of gene expression enables bacteria to respond to changes in the environment, particularly the presence or absence of nutrients. The rate of synthesis of gene products can be regulated in three possible ways, namely by regulating the rate of transcription, by regulating the time of mRNA turnover, and by regulating the rate of translation. Of this, transcriptional control of gene expression is the most significant and the best understood one. So, transcriptional control of gene expression in bacteria. Transcriptional control of gene expression in bacteria can be intrinsic and extrinsic. In the former, internal factors control transcription, and in the later, external factors are involved. So, intrinsic regulation involves the control of the transcription of specific genes by a repressor protein specified by a regulator gene. This protein decides how strongly the RNA polymerase can bind with the promoter gene. This in turn decides how often transcription can occur. Genes with weak promoters are usually transcribed at low rates. In fact, they are the genes whose products are needed only in small amounts. The weak promoters minimize the expression of genes. On the other hand, genes with strong promoters have high rates of transcription and their products are needed in large amounts. In extrinsic regulation, the rate of gene transcription may be turned up and down by factors that are extrinsic to the gene. Extrinsic regulation is commonly among genes with strong promoters. Then another major topic like operon concept. The transcriptional control of gene expression in bacteria is best explained by the operon concept advanced in 1961 by the Nobel laureates Francis Jacob and Jacob and Monod, based on their brilliant work on Escherichia coli. In a sense, it is the concept of the coordinated regulation of genetic transcription to bring about the repression or de-repression of gene expression or the inhibition or stimulation of enzyme protein production. Operon concept upholds three basic assumptions, namely induction and repression of gene expression are accomplished essentially by the same mechanism. At a time, only the required enzyme or proteins are produced in the required amounts. The production of an enzyme or protein is induced when it is needed and inhibited when it is not needed. Usually, it is the presence of the substrate that induces or triggers the production of new enzyme molecules. The substrates whose presence induces enzyme production are known as inducers. Most inducers are then metabolized by their own enzymes, but some are not, and they are called gratuitous inducers. The enzymes whose production is induced by the presence of substrate is known as inductive or inducible enzymes. The genetic systems which govern their production are called inducible genetic systems. In some cases, the presence or addition of the end product of a synthetic pathway can repress the production of the enzymes that are essential for its own synthesis. Such end products are called repressing metabolites or co-repressors. Some amino acids such as histidine. The enzymes whose production is repressed by co-repressors is known as repressible enzymes, and the genetic enzyme systems which govern the production of these enzymes are called repressible genetic systems. Then, inducible and repressible systems of genetic regulation. According to the operon concept, there are two systems of genetic regulation in prokaryotes, namely the inducible system and the repressible system. The former exerts positive and the later exerts negative regulation. Inducible system operates when the substrates are available and enzymes are required to metabolize them. On the other hand, the repressible system operates when substrates are not available and enzymes are not required. It may also operate when the end product of a metabolic chain are far in excess and not required any further. Repressible system consists of a regulator protein called a co-repressor. It is not capable of binding with the DNA all by itself, so it combines with another molecule called co-repressor and forms an apro-repressor co-repressor complex. This complex represents the active repressor. It binds with DNA and represses genetic transcription. Then organization and operating of a generalized operon. An operon is a group of closely associated and coordinately regulated prokaryotic genes functioning together as a single transcriptional unit in gene expression. Its genes act in close coordination and mutual regulation for the production of enzymes of a metabolic pathway. In general, an operon consists of one or more structural genes, an operator gene, and a regulatory gene, and a promoter gene. Structural genes code for mRNA and enzymes, so they direct the synthesis of enzymes through transcription and translation. If a few structural genes are present, they transcribe either separately and independently to form a few monocystronic mRNAs or together to form a polycystronic mRNA. Operator gene serve as an on and off switch of the structural gene. It is located close to the first structural gene. It constitutes or activates the structural gene only when a specific enzyme is needed and represses or inhibits them when the enzyme is not needed. So, operator gene plays a significant role in enzyme production and metabolic regulation. The functioning of operator gene itself is under the remote control of a regulator gene located far away from the operator site. The regulator gene codes for a repressor protein. This protein directly represents the operator gene and thereby indirectly inhibits the expression of structural gene and the production of enzymes. At times, when the enzyme is not needed, the regulator gene undergoes transcription and guides synthesis of repressor. The repressor recognizes the operator gene, binds with it, and forms a repressor operator complex. This complexing locks the operator gene in off position, so operator gene fails to activate the structural gene to undergo transcription. Repression of transcription occurs by blocking the RNA polymerase by the overlapping of the operator over the promoter. So repressors are capable of binding not only with the operator but also with inducers and co-repressors. They exist in active and inactive states. They are active when they remain bound with co-repressors and inactive when remaining bound with inducers. In other words, binding with inducers inactivates the repressors and binding with co-repressors activates them. Binding of repressors with inducers or co-repressors occur when weak bonds such as hydrogen bonds are produced. So strong associations like covalent bonds are not formed, just weak hydrogen bonds are there. Repressor-induced complexing causes conformational changes in the repressor. As a result, it loses its ability to bind with the operator. This in turn unlocks both the operator and structural genes and keep them in active functional state. On the other hand, co-repressor complexing promotes repressor-operator complexing and thereby represses the functioning of the operator and structural genes. Promoter gene is contiguous with the operator gene. It contains a signal codon that indicates the binding site of RNA polymerase and specifies where transcription of the structural genes should actually begin. Operons function for both repressing and inducing gene expression. Accordingly, two kinds of operons can be recognized, namely repressible operons and inducible operons. In the former, the repressor protein is in active state and it remains complexed with the co-repressor, but in the later, it is inactive and remains complexed with the inducer. Inducible operons contain genes which encode catabolic enzymes. Expression of these genes is controlled by a substrate of the catabolic pathway. Lactose operon is an example of inducible operon. It encodes the enzymes that are necessary for lactose metabolism. Repressible operons contain genes which code for biosynthetic or anabolic enzymes. Then expression of these genes is controlled by the end product of the pathway, which may either repress the operon or control it by an alternative mechanism called transcription attenuation. Attenuation is the regulatory mechanism by which the transcription potential of an operon is transitionally coupled to specific control regions of mRNA. Attenuation is a regulatory mechanism by which the transcription potential of an operon is transcriptionally coupled to specific control regions of mRNA. Functionally, it serves as a mechanism for regulating the expression of bacterial operons which code for the enzymes of amino acid biosynthesis. Tryptophan operon, TRP operon is an example of repressible operon. So, attenuation is a regulatory mechanism by which the transcription potential of an operon is transcriptionally coupled to specific control regions of mRNA.